So the complement system is a part of the innate immune system. Complement are proteins that are produced by the liver and then secreted into the bloodstream, which then are allowed to circulate so that upon disruption of homeostasis, such as invasion by a pathogen, the complement system will be one of the very first things to respond. And so just to quickly cover some of the important aspects of the complement system, it is part of the innate immune system, so it's going to be very rapid, and it will actually play a really big role in lysis of cells, whether bacterial or viral particles. So it will kill them, but it will also, I have it down here on the third point, but it will inhibit the pathogen by blocking essential proteins. So, for example, if you have bacteria, they are, in order to establish infection, most of the time they're going to need something to adhere to, basically to target cells, and complement proteins can prevent that from occurring by basically coating the surface of the pathogen. And so this coating serves two purposes, opsonization being the second one. So the first one is it can inhibit or kill the pathogen, and then opsonization of the pathogen so that you can promote additional immune cells from both the adaptive and the innate immune system. Okay, so there are three ways to activate the complement system. You have the classical pathway, the alternative, and the mannose binding lectin pathway. So the classical pathway, let's separate these. So the classical pathway is activated by the recognition of antibodies, so antibody antigen complexes. So I will antibody antigen complexes. So whatever the pathogen of interest is, eventually or somehow in the body, you will have antibodies that are capable of recognizing the surface of it. And as those accumulate, the constant region of the antibody will promote the complement system. And so it starts with C1, which will then bind to this part of the antibody. And then when you have C1, this initial binding is what starts the complement cascade. So remember, these are just proteins that are floating around in the body. In order to be activated, most of them require cleavage from the preceding member in the pathway. So what I mean by that is basically without the starting protein, everything downstream can't happen. So if C1 binds to the constant region of the antibody on the antigen complex, this will promote and cleave a protein called C4, and that will cleave it into two separate proteins. So it'll be C4A and C4B. And as I'm writing these, remember, I'm not interested in your memorization of these numbers and subunits and things, but I'm providing a little bit more detail so that hopefully it provides enough context so that you can use this when asked about on evaluation. Okay. So the active portion now that this has been cleaved into two pieces, this becomes the active portion, and it will convert C2 into C2A and C2B. C4B, C4B and C2A combine to form what's called C3 convertase. Okay, so this enzyme is the combination of two previously inactive complement proteins. So it all started with C1, and then if C1 is active by binding the antibody, it will cleave C4, which cleaves C2, and those subunits combine to create a new enzyme. Not surprisingly, C3 convertase is responsible for cleaving C3 into C3A and B. C3b then, once this is cleaved away from the original protein, will combine to C3 convertase, and all of that will become C5 convertase. Sorry. So remember, this is made up of, if you would like to follow all of the active pieces, it's made of C4b, C2, so I'm only going to write C once, sorry, C4b, 2a, and 3b. And then this is what is responsible for promoting C6, C7, C8, and C9 to the surface of the pathogen. So remember, it all starts on the constant region of the antibody, but then as things happen, they bind to the surface of in this case, we'll say it's a bacterial cell. And then C5, 6, 7, 8, and 9 form the membrane attack complex. So write it like this. I'll just abbreviate it, the membrane attack complex, remember MAC attack. And then it is that which forms a little pore, or I guess a rather large pore, on the surface of the pathogen. Oh, I'm sorry. This is hard. Okay, that is supposed to be a pore. Okay, and so remember this complex, because it's part of the membrane now, it has integrated itself into the pathogen's membrane, made up of these 5 proteins. I guess C5 is also on there. So these 5 proteins. And then once this is formed, it's just going to, the cell is going to leak out, it will depressurize and leak out its insides, which then allows for recognition by additional immune cells. The alternative pathway, it will follow a really similar cascade style. The only difference is how the cascade begins. So in the alternative pathway, the main or the only initiation signal is going to be the presence of endotoxin. And so remember, endotoxin refers to surface components on gram-negative bacteria called lipopolysaccharide, I think I spelled that right. Also known as LPS for short. So in this pathway, rather than beginning with C1, C3 is capable of recognizing this on cells or in blood serum. Because remember, as bacteria divide, at least as gram-negative bacteria divide, this is being shed into the external environment. And so this will spontaneously bind to LPS, and then it will cleave into C3A and B, similar to the classical pathway. And then there are additional factors. I'm not going to include them, but they are called factor B and factor D, which follow, again, it's a really similar pathway. Factor D will cleave factor B. I will just write it. So once C3 binds it, C3b will promote factor D. Sorry, I don't know what I'm doing. Factor D, which then allows factor B to be cleaved into B, B, and BA, which then will go back to a really similar cascade to the classical pathway. And then once this is active, it will essentially help C3, the original C3, to become part of the C3 convertase enzyme, which then will activate these proteins down here, and it will still lead to the MAC attack complex. This is always, in all three pathways of complement, this is the end goal, because this is what allows the intracellular components to be exposed, and it will kill cells. And the last pathway, the mannose binding lectin pathway. So this is a protein also present in the blood, and this will specifically recognize sugars present on certain bacteria and some yeast. It is going to be the least used of the pathways. So basically, I just want you to remember that it's a sugar binding pathway. These sugars being what are found on the pathogens. And then it will also initiate a cascade from the complement system, all of which, again, because this is the end goal, it will lead to C3 convertase, which then converts into C5 convertase, which then leads to the pore formation using these 5 proteins. Okay, couple last things about the complement pathway. Our body has to protect itself from this cascade initiating, because there's always going to be some level of antibody-antigen complexes occurring within outside of our own cells. And so in order to prevent that from happening, our own cells must have what's called factor I. You can remember that I as in like me. If a cell has this, then it will prevent this cascade from proceeding by blocking the cleavage of C3 into C3A and B. If this does not occur, then C3 convertase cannot become C5 convertase, as it requires the C3b subunit. So by essentially coating this protein, it releases it from the surface of the cell and prevents this pathway from occurring. In B cell maturation, if there are antibodies that target our self, then these antigen-antibody complexes will still occur, and there are actually autoimmune diseases associated with activation of complement on our own cells. And so, just to finish up, so this is my little human. If we have, okay, so apparently the hand's been cut off, so there's going to be some pathogen entrance into the body this way. So the first thing to initiate is going to be the complement system, and that is going to lyse bacterial cells, which is going to lead to, again, leakage of intracellular components. At that point, then you're going to have phagocytic cells that come along and engulf these pieces, Pac-Man style. And then they will travel then up the lymph system to the lymph nodes. So these dark spots, dark green spots, are just some of the locations of the lymph nodes. There are more than this, but I just wanted to kind of show you where they were. It very closely resembles the circulatory system. Essentially anything that is found outside of the circulatory system and is fluid, it's going to flow through the lymph system towards the heart, just the same as your blood. The difference being that it will congregate in these lymph nodes at different locations where there will be really high concentrations of antigen-presenting cells. So most of the antigen presenting cells in our body are actually going to be found in any of the lymph nodes at any given time. Only 1 to 2% of our immune cells are circulating through the bloodstream at any given time, which means most of them are going to be at these locations. The reason that they congregate there is because that's going to be the most efficient way to present antigen and get activated by T cell. There is another video that talks about T cell activation. Like I said, because most of the cells are going to be in the lymph nodes, that is going to be where the most likely place that you're going to find a T cell that also recognizes your same antigen to activate you. And so, remember, this all can initiate with a complement system that leads to bacterial cell or viral particle lysis, and then leakage into the extracellular environment, where it can then be engulfed by phagocytic cells, which tend to also be antigen presenting cells. So in this case, it's a macrophage. So the macrophage will then travel through from the site of infection to the lymph node, where it happens to find a T cell that recognizes it, and then will initiate an immune response in this way.